Let's just take a look at the difference between prokaryotes and eukaryotic cells. So the big obvious difference is that prokaryotes do not have a nucleus and there are no organelles. And then eukaryotes obviously do have a nucleus and they do have organelles. And another difference is that in general, prokaryotes are going to be much smaller in size and less complex than the eukaryotic cells. And eukaryotic cells are generally going to be larger and more complex. And another difference would be with their chromosomes. Prokaryotes usually only have one chromosome, and the shape of their chromosome, it's usually circular. So they usually just have one circular chromosome. And that's really different from the eukaryotic cells. Eukaryotic cells are going to have two or more chromosomes, so it just depends on the species. So for example, like we have 46 chromosomes. And the shape of the chromosomes are going to be linear, so they do not make a circle. And another difference would be the size of the ribosomes. Prokaryotes have what are called 70S ribosomes. And eukaryotic cells have 80S ribosomes. And the S just refers to a unit that's used to measure when they centrifuge things, just at the level where they centrifuge out. It's called a Svedberg unit. So the eukaryotic ribosomes are actually bigger and heavier than the prokaryotic ribosomes. The reason this is important is because there is a class of antibiotics that will only bind to the bacterial 70S ribosomes, but they won't bind to the eukaryotic 80S ribosomes, which our cells have, which is good. Okay, and we'll learn about those later. Okay, and another big difference is how prokaryotic cells and eukaryotic cells divide. So prokaryotic cells are going to divide by something called binary fission. So they do not do mitosis or meiosis. So that does not happen in prokaryotes. And then eukaryotes will divide by either mitosis or meiosis. Remember, meiosis is the one that's used for sexual reproduction. Okay, and then here's just a nice picture that just shows the difference in size. So these two large cells here are eukaryotic cells. These are epithelial cells. And you can see the nucleus in each of the cells here. And then here are some prokaryotic cells here, some bacteria. So you can see there's a huge size difference between prokaryotic cells and eukaryotic cells. That's a nice visual there. Okay, and we're just going to be focusing on the prokaryotic cells here. So let's take a look at the two domains that prokaryotic cells are divided into. So remember, they're divided into domain bacteria and domain archaea. And we're going to be focusing mostly on domain bacteria. So the bacteria are going to be divided further into two groups based on the Gram stain. And the Gram stain here is pictured on the bottom. We've got the bacteria dividing into Gram positive, which stain purple, and then Gram negative bacteria, they stain a red to pink color. Okay, next we're going to take a look at the bacterial shapes, and there are three basic bacterial shapes that are really important to know. So the first one is cocci, which is plural, and the singular would be coccus. And this shape, the bacteria are spheres or little round balls. Second shape would be bacilli, and that's plural, and then bacillus would be the singular. And these are rods, and so they would have a shape. Oops. Looks like that. And then our third shape would be spiral, and they're going to be curved. So they would look like that, or maybe go over here, or maybe they look like that. Okay, so they're curved in some way. Okay, and then so these are the three basic shapes. So when you're looking under the microscope, you just want to see, are they cocci, bacilli, or are they spiral? And then we can look at their arrangement after that. Okay, so here's our bacterial shape and arrangement. So we've got our three basic shapes. We've got the cocci, bacilli, and spiral. And then the bottom we have in blue pleomorphic, which just means that we do have some bacteria that will vary in shape, and that's because they don't have a cell wall, so they can kind of change shape just depending on their environment. But most of bacteria do have a cell wall, and so they will be one of these three basic shapes. The arrangement is just talking about that when bacteria divide, sometimes the cells remain stuck together. So, for example, with our basic cocci shape, if the two cocci stay stuck together, we would have diplococci, because diplo means two, and cocci, you've got the little round balls, the spheres, so that would be a diplococcus, okay, when you have the two spheres stuck together. Streptococci, strepto means chain, and so you'd have a chain of cocci together. Staphylococci, staphylo means grape-like cluster. So you would have the cells looking like a bunch of grapes. Okay, and then a tetrad, tetra means four, and so you'd have the cocci, four of them together forming a square. And then sarciny, that just means cluster, and that's where you have 8 of these cocci, and I can't draw this one really well, but you would have 8 of them together forming a cube. So sarciny, that's kind of a strange word, but it means cluster, and you end up with 8 of them making a cube. Okay, and then the bacilli are rod-shaped. You could have a single bacillus, just one of them by themselves, or you could have diplobacilli, where you'd have 2 bacilli joined together like that. And then you can have a chain of bacilli, so streptobacilli. And then the last one is kind of strange, coccobacilli. So these guys can decide if they wanted to be a coccus or a bacillus, and so they're kind of a cross between the two. So they're a short rod that's rounded on the ends. So that's kind of what they look like. Okay, Bordetella pertussis is a coccobacillus, so that's an example of that one. Okay, and then we have our spiral shapes. So remember, these are curved in some way. A Vibrio, they're a curved rod, so they look like a comma shaped. Okay. And then a spirillum, that's the singular, and spirilla would be the plural. These guys are spiral shaped, and whoops, sorry, that wasn't a good picture. But they are thick and rigid. And what we mean by rigid is if you're looking at them while they're alive under the microscope, they're not able to bend as they're swimming around. Okay, so you won't see them able to bend and swim around the corners. Okay, and then we have the spirochetes. So these are also spiral, but they're much thinner than the spirilla, and they're flexible. So as you're watching them while they're alive under the microscope swimming around, they can actually bend, so they can kind of bend around the corners. Okay. All right, so let's just take a look at some more pictures. And up here at the top left, you can see we've got some diplococci, so 2 spheres together. Here's streptococci, chain of cocci there. Here we've got a tetrad, 4 making a little square here. And then sarciny, you've got 8 cocci making a cube. Okay. And then staphylococci makes the grape-like cluster. And then over here with our bacilli, the rod-shaped, we can have a single bacillus, like some of these over here. And then the diplobacilli would be 2 bacilli joined together, which there's kind of some of those over here on the far side. And then streptobacilli would be a chain of bacilli. And then here are these coccobacilli. So there are these short rods with these rounded ends. So they couldn't quite decide what they wanted to be. Okay, and then here's some examples of spiral bacteria. Here's the curved rods. So they look like little commas. So those are Vibrios. And then here's a spirillum. So they're thick and rigid, and I would tell you that. So they're not able to bend and go around corners as they're swimming. And if you can compare that with the spirochete is thinner than a spirillum, and it's also flexible, so you will be able to see it bending as it swims. So it could bend here in the middle and go around a corner if it needed to. Okay, and then just kind of a question here. Think about what type of microscope took this picture of this spirochete. I'll give you a hint. If you notice that the background is dark and the specimen is bright. So hopefully you were thinking about a dark field microscope. So dark field microscope took that, and the advantage of that type of microscope is that it increases the contrast without staining. So you could look at this living spirochete without having to stain it, and so you could see it swimming around, and so it's still alive. Okay, so what I would do on the test and the quiz is that I could give you some pictures, and then you have to identify the bacterial shape and arrangement. So if I gave you this top left picture, you would identify that as streptococci. If you just gave the answer strepto, or you just gave the answer cocci, you'd only get half credit. You have to give the full answer, streptococci, because you've got this chain of cocci. Okay, and then over here on the right top here, these would be some grape-like clusters of cocci, so that would be staphylococci. And then right below here on the right, this one would be chains of bacilli, so that would be streptobacilli. So the answer to that one would be streptobacilli. Okay, and then the one on the bottom left, I would give you more information and tell you that this one is thin and flexible. So hopefully you came up with the answer of a spirochete on that one. So spirochetes are thin and flexible. Okay, and lastly, I just wanted to show you this picture to show you size difference. So we can see a couple different shapes in here. So here's some bacilli in here. They have labeled as rods, but we would call it a bacillus. And here's a diplobacillus there. And then notice the size difference between the coccus and the bacillus. So cocci are much smaller than the bacilli. And then we also have some vibrio in here too. So these little comma-shaped looking ones. Okay, so those are vibrio.